So, hey everyone, thanks for having me. Really excited to do this. I think there'll be opportunity for more technical talks as more of our PRs get merged and go through, diving into like deep decisions and implementation details we've made through either existing or future features in Flyte. But I wanted to take this opportunity to really walk through like what we're doing at Latch, how it touches Flyte in many different ways, and how workflow orchestration is even motivated by needs in biology. So yeah, just diving ahead. About us, we build software and data infrastructure for companies in biotech. We're at this point a 10-person company founded out of Berkeley, and this is the founding team in all of its glory. So really quick, wanted to dive into some trends, motivating, and also just keeping tabs here. It's like roughly 20 minutes, right? Tom?
Yeah, I think you can go 20, 25 minutes. That's fine. Just go for it.
Great. Yeah, so just some trends in the space. Like this is a corollary to Moore's law, which everyone has seen. As the constituent components of computers get cheaper, you form these classes, these computing classes that also get cheaper. And this is going to be analogous to some things we see in biology, but this graph just basically shows all these classes of things. Their price goes down over time and their accessibility goes down over time. And then similarly, and particularly relevant to workflows is the performance of file systems and IO operations per second and throughput over time, as well as capacity. And this, it goes by the canonical postulate of common sense, as it were. So the analogs in biology is that the primitives are getting cheaper to read and write. Everyone loves throwing around this graph. It's the cost per genome, the cost per base pair for sequencing. And you can see it's rapidly exceeding the rate of Moore's law. Similarly, writing DNA, so synthesis is getting far cheaper. In fact, the rate at which we can write or synthesize novel pieces of DNA is exceeding that at which we can read it. This is significant because we can now like write genetic code that did not have an origin in nature. We don't have to. Molecular biology in the 1970s and 80s relied on taking components of pre-existing logic from bacteria, from existing organisms, and stitching them together in new ways. What DNA synthesis allows us to do is completely specify new logic arbitrarily and see what the heck it's going to do in a living system. So these trends closely mirror what we saw in computing, and the fact that they're exceeding the rate at which computing progressed points to some interesting things. So let's look at a couple more foundations for Latch. There's this field called synthetic biology, which was founded and coined in the early 2000s out of Tom Knight's lab at MIT and Drew Endy's lab at Stanford. And the idea is that we should reduce biology to constituent building blocks and apply engineering principles to make de novo life from scratch, right? So an early initiative was called the BioBricks project, where we can take genetic components, treat them as Legos, and rebuild them, even rallying kids in high school and college around competitions to build the best model organism. It's kind of nutty, but it's happening, it has happened, it's continuing to happen. So we can translate logical genetic components into like things that exist in living organisms. Promoters are, constitute how you would start or stop a gene and the logic thereof. You have repressors, which are like equivalent of like an off switch. You have the things that the genes are expressing. You have the terminators which stop them. You can imagine mixing and matching logical components to produce behavior that you want in a living system. So this is the rise of synthetic biology. And from this building philosophy, we've had many amazing things, one of which is the rise of metabolic engineering. Most of our biological foodstuffs, so as well as drugs, are produced from enormous fermentation systems. This right here is Amyris, located in Emeryville, California. And this is an example of how they've taken this philosophy to production scale to produce things that we care about and buy. And in addition to that, there's really interesting theoretical academic applications of this in genetic circuitry. So you can make really, really complicated logic from basic genetic constituents. So that's kind of the philosophy, the thought shift in biology that is the tailwind at which Latch is resting. Now we have the tools. So in biology recently, there's been some like really interesting ways to measure stuff. This is called flow cytometry. And what this means is you can basically take cells, shoot them with high fidelity through a laser beam, and sort them into bins based on properties of those cells with the diffraction of that laser beam interacting with some sort of sensing system. This means you can basically make changes to millions of cells at a time and look how they're going to behave on a single cell resolution, collect them, and then grow them and read them out downstream. It's very cool. And then this is an example of what we call like a multiplex high throughput library. In biology, a library is just a large collection of genetic designs. And so that it obviously is compounded by the decrease in cost for DNA synthesis. It is also kind of compounded by the rise in gene editing technologies over the past decade or so. I'm sure everyone's been following or at least heard of Jennifer Doudna at Berkeley and the whole war between Berkeley and MIT and licensing gene editing technologies, but they let you edit base pairs or single nucleotides in a DNA sequence with incredible fidelity and programmability. Right. And so the last thing here is the market. So like, who the heck are we selling to? Over the past 10 years, spearheaded by all these technologies, is the rise of what are called cell and gene therapy companies. Now, cell therapy is where you take someone's own immune cells out of their body, re-engineer them, usually with a CRISPR-mediated viral vectors. So you're using viruses to inject CRISPR into the cell, re-engineer its genetic code to an extent, re-express some sort of protein that's going to kill cancer. Using this kind of modality, companies are developing what's called off-the-shelf cell therapies to cure people of cancer. And then the analog, or not analog, but closely adjacent is a gene therapy, which is where you directly inject the patient with a CRISPR-loaded virus. And then that CRISPR-loaded virus targets tissue with high specificity and is able to cure people with disease. So this is who we're selling to. And these guys are blowing up like crazy over the past 10 years as this technology is becoming cheaper, more accessible, and it's being augmented by computation. So let's get into computation. The last piece of the puzzle is the nature of biological data. It's really big, it's really heterogeneous, and it's really hard to interpret and wield. This is an example of a tiny chunk of a genome, and these genomes on average are gigabytes in size. The human genome is 3.3 gigabytes in size. The spruce genome is like 15 gigabytes in size. This stuff is unwieldy and hard to wrangle. In order to wrangle it, you need to take these short reads from a sequencing machine, kind of align them, and interpret the consensus of a bunch of short fragments. This process takes days. It takes dozens of cores. It takes hundreds of gigabytes of memory. And this is on a per sample resolution. If you're doing any of these experiments that we talked about earlier, you're gonna need to do this dozens, hundreds of times, right? And from doing this kind of assembly, this is kind of facilitated by algorithms you might all recognize, dynamic programming and Bayesian graph facilitated reconstruction. You can generate, interpretable information about how cells are structured, where they're grouped. You end up taking like vectors about like what kinds of genes are being expressed in different types of cells, and you can understand things about how diseases are progressing or how well your treatment is working. And additionally, you can say things about like how well CRISPR might be editing your cell. These are just two examples of how you might interpret what is otherwise completely uninterpretable data that would need some sort of computation to make into something you can understand. Right. So I thought, I would just show you all a quick video of what we're building. Our platform is designed to enable this computation at scale with heterogeneous biological data for the types of companies I showed you. And the way we do that is relying on Flyte's workflow execution engine to, have highly scalable Kubernetes native type-safe workflows. And we generate no-code interfaces for the biologists to use directly, dynamically from a Flyte kit wrapper. And by doing this, we can expose a myriad of different bioinformatics tools to the end user and provide a really rich in-browser suite of visualizations and file manipulation tools. So this is our platform. And then on top of the platform, we're starting to tap into the bioinformatics, computational biologists, and even software engineers at companies and giving them their own toolkit to both write Flyte workflows and dynamically generate Latch interfaces on top of them. So diving into a couple of technical details of the platform, the first component is a managed data store. And we really recreated pretty much a fully featured Nix flavored file system in the browser. RAS arbitrary file stores, at the moment it's just S3. But the moment you can move these around, drag and drop them, and visualize them, see all that metadata you want in a much more convenient way. That's like the tip of the iceberg. The moment files with semantic file extensions hit the system, we run workflows as hooks on top of them. A great example is a DNA file that we recognize. If you upload it to the platform, we run a Flyte workflow automatically to parse, run quality control, and generate visualizations on top of that file, such that when you double click on it, you can then see those visualizations, right? Additionally, because we have our own file system, we can support our own network protocol and our own absolute path system. So you can pass these object files, objects or files to workflows directly as semantic paths, and the workflow logic and the Flyte wrapper will parse these and understand them, making it much more easy to reason about where your data is coming from and where it's going and how you can manipulate it downstream. We found that, I'm sure you all have found that as well, that S3 and bucket stores are incredibly unwieldy. When working with large scale projects, it doesn't feel like a file system, and you really need that to scale things out. So on top of the file system, we have compiled type safe UIs. And what this means is we basically take the parameter interface from Flyte ADL, and we dynamically parse this and compile React interfaces from the typing information. Now, from typing information, we can construct components with in-browser HTML native type validation that makes sense, as well as slap on rule-based regex validation on top of HTML native validation to add like rich layering of browser side type checking before the workflow even hits Kubernetes. So there's a lot that we've done here. We have a really talented designer and a really talented product team that's really like watching how biologists interact with interfaces, and we want to make sure that workflows don't fail before the programming logic gets executed in the container on the cluster. And more to talk about here in a sec. So the last component here of the platform is the serverless scheduling, fine-grained scheduling, and why we think Flyte is so important and why we think Kubernetes is so important. That's just grounded in two distinct biological use cases. So the first reason why we need Kubernetes and Flyte is we want per task control over how we schedule tasks. And this is because we might have entirely different needs for an upstream or downstream task in both compute environment and resources. A concrete example is we want to do one of these genetic assembly operations on a highly threaded machine. And then we want to feed the resulting assembly to a GPU-enabled machine for inference on some sort of ML model, maybe pick out components of the code that are relevant towards some experiment. Right? It's a very concrete example of why we need something like Flyte. Another reason is obviously scalable scheduling. So like if you need to do one of these genomic assemblies on a human genome, again, 3 gigabytes, it'll take you 2 days on a machine that costs a lot of money to run. If you can leverage spot instancing and automatic retries through the kube scheduler and scale this out over Kubernetes, finding, relying on the scheduler or like a cloud-enabled scheduler to find capacity as it comes up, rather than implementing yourself on bare metal or something like that, you can really do this thing at scale in a way that other platforms can't. So this is the rough architecture of our system and how Flyte slots in. We have a core Kubernetes cluster, which supports a bunch of services. It's not super interesting. It's pretty standard web stuff outside of the fact that everything's Kubernetes native, including not just Flyte. We have a bunch of services named after biological types that maintain a set of local state that is tied to drop-in Flyte deployments with our own scaffolding called Prion in private VPCs, either in customer VPCs or in our own managed VPCs. What this lets us do is maintain a proxy file system and proxy workflows without actually ever touching the data or the workflows of customers, but puppeting them around in remote systems using a two-way sync service called Ribosome. And this is really hard to do, as you all can imagine. Engineering team has spent a considerable amount of effort like getting this right. There's still things we need to work out to make this scale, but essentially it's lots of queuing and lots of SNS notifications from the AWS side, as well as updates from a serverless system on the Ribosome side. Stateless system on the Ribosome side. There's basically queues on either side of any sort of file movement or workflow update propagation, and that lets us like keep a proxy of really unwieldy objects in local state and keep console snappy. That's roughly how things slot in. And again, the biologists interact with the platform via the console, and the computational biologist interacts directly with a server via the SDK, and everything comes from the database in terms of like state hydration. Where only 5% or so of the requests are synchronous through the HTTP server. So jumping in really quick, why Flyte? Again, so it's K8 native, and so in conjunction with EKS or some sort of cloud-hosted solution that it takes care of provisioning instances for you, we can provision any computer we want, which is incredibly important for what we want to do. There's language independent type safety. We wanted to support all the type primitives that exist in canonical programming languages, as well as expanding biological types, so we can support more fine-grained checking of data things through workflows we'll show you. And we also thought the language independence was important because developers of bioinformatics work with all sorts of languages, although Python is the most ubiquitous at the moment. And then I would just explain tasks as independent deployment, incredibly important for what we do. And then we wanted a open source and well-maintained code base that we can understand, interact with, work with the team, and push improvements to the general community. So far we've made several contributions, and we have a lot more planned on the way. We're working on some stuff in-house that we want to spin out. But our contribution velocity and the rate at which we're contributing is a reflection of like our confidence in Flyte long-term as the de facto workflow orchestration engine. I'm not just saying this, I really think Flyte has got the model correct, absolutely correct, with respect to how you're thinking about architecting and deploying workflows as both code first and leveraging the community scheduler to abstract away the scheduling on a per task granularity. Really think that's the absolute right way to think about it. And so we want to help support and grow this project and make sure it sticks around for a really long time. I really think it'll be around as long as Docker and Kubernetes. We've contributed typing metadata, Docstring metadata, and Max is still finishing up union types. But we'll dive into Docstring and typing metadata in a second. Union types is, as you would imagine, optionals, some between none and some other arbitrary type, which is really important for us to make sure that we can catch null none values before they hit the workflow. So really quick, why do we want parameter metadata? What is it? I talked previously about adding server or client-side regex role-based validation on types. So if you want to add information to change how a type is parsed on the front end, again, considering that we completely generate our interfaces from the function header alone, you need to be able to stuff arbitrary JSON into the type literal in the language at which you're developing. So in our case, talking about Python, we want to annotate the Python type with a class, but let's just like add arbitrary information so we can parse that on the front end and tell the user, hey, your string doesn't actually conform to the subset of strings that we're allowing in this case. Nucleotides can only be A's, T's, C's, and G's. If you add an X, it's probably incorrect here. We want to display that information. So by defining a type, exposing it to the user either through the SDK and like kind of a predefined stuffed value, and allowing them to annotate the parameter in their function, we can then generate these parameter components that do exactly as I just described and are incredibly pluggable and let people do this on their own if they want. So this also extends to biological types. If you want to annotate like a file extension with, to make sure that if this workflow is only taking this type of file, we also do the exact same thing with the same sort of logic. And you can find this supports almost anything you want. Really quick, we also have Docstring metadata, and this allows us to define kind of the layout ordering display of a workflow interface completely in a function Docstring, borrowing from frameworks like Sphinx and how documentation is parsed from restructured text in those Docstrings. But everything from ordering to the display names to groupings to hidden parameters to tooltips. Later, actually this week or next week, paired groupings of parameters that must have certain state in accordance with each other can be enforced in the Docstring of the workflow itself. And this is really creating kind of a new paradigm, which we're calling like interfaces as code on top of Flyte kit, which allows you to completely define front end in like a couple of Python files and version control, which I think is very powerful. And so lastly, to wrap things off, wanted to show examples of like an internal modification we've made to the Flyte system. I mentioned we have this Latch network scheme, which allows us to resolve and understand file objects with respect to the absolute path for any given user's working directory in their account. And we do this by proxying requests for files within Flyte kit to manage endpoints to authenticate and like then serve the pre-signed URLs to the task code. And we actually met with the Flyte team to figure out the best way to do this. We're using the execution name to store secrets, and it's like a really cool and secure way to do something like this. An example of like how we've leaned on the Flyte team to develop something that worked for us well. And so lastly, I want to talk about another big feature we're working on in-house and are excited to release and work with the Flyte team to integrate into the master code base is server-side containerization. Constructing Docker containers locally is a bad experience for a lot of people, and it's expensive and can be improved drastically, right? Oftentimes you have to pull large base images. Oftentimes people don't even know how to use Docker, which for some is surprising, but that's like really often the case. And then you have to pull large layers. They use lots of space. They use your local file system throughput, your CPU. You can't even watch like YouTube while you're building a Docker container. And you have to push the containers back up. It's terrible. So we can create a specialized container build service. We're calling Centromere, and we'll release this open source in the coming months. But by reusing, very naively, if you just think you can reuse a shared file mounts and shared build environment to keep a warm cache of file layers and prevent the network transfer of the build context to Flyte and just do the registration on the server side. There's something more we can do in a second here, but like this is the basic idea. Any sort of local state you need, you can parse from the Docker file and just send those individual files up to the said service and use that to build the Docker context, the Docker build context. One additional thing you can do, if you want to like roll out unilateral configs, you can recognize that a layer is really just file archive with the metadata and hash. And so if you want to change something like, the value in a config file, like your secret in a flyte.config file or something like that, you can just pull apart the archive, manipulate the file, slap it back together, and make sure the hash is the exact same. And as long as you like understand that your change to the aggregate file layer is safe, you can then deploy this unilaterally to all the containers that your users use. So this is another interesting idea we're exploring and playing with. And so this avoids triggering rebuilds for really big workflows. And so that's it. Happy to talk about, I know this is like not it was super deep, and we're looking forward to doing maybe a deeper one later on some of existing or future contributions, but this is like what we do and how we're using Flyte. So that's it.
Thank you, Kenny. That was fantastic. I'll ask, there are a bunch of, I think, biotech people here, so I'll open up questions.
Kenny, I'm curious what you think about handling like private data or PHI or protected data on Latch for like public accession and like making sure that analysis is reproducible, on large scale projects, similar to like Cromwell or other technologies.
Yeah, I mean, so we, like I explained, the way our architecture is set up, maybe I'll just pull that up again, is that we can really address all of these concerns about holding on to the actual files or holding on to actual workflow state by having Flyte deployments and completely closed off VPCs and puppeting their execution and puppeting the movement of files without actually touching them. So again, what we do is we hold references to the location of the objects, and we hold references to the workflows themselves, but we never actually hold on to them within infrastructure that we own. So that's one way we deal with this. And this is an incredibly hard thing to do, but it's something we've done for the exact reason that our customers are pretty sketched out about this. We also are mostly preclinical. So on the discovery side, pre-IND, and we don't deal with any API or any clinical information at the moment. Yeah.
Gotcha. Thanks.
Absolutely. Yeah, and also like.
Oh, sorry. Go ahead.
I was just gonna say, if you guys are going to talk about, if you have experience in biology, I want to talk about that as well. Our team has experience there. I'm happy to talk about that.
I also have a related question to the metadata you store on your side. I see that it's like Flyte pointers and things like that. Do you keep like workflow names, the spec of the workflow, the task names, primitive inputs, like these kind of data, do they live, or do you copy them in your cluster, or do they stay exclusively in the user side?
Yeah, so all we hold on to is like the typed interface, and that's what we need to both generate the front end and also to make any sort of like, actually create the execution through the Flyte control service.
Okay.
And then, yeah, I mean, outside of that, it's metadata about the execution name when it starts and ends, as well as references to the location of the object, if any files are being passed in. But that's the extent of it. As like, we have full Flyte deployments set up with, I think, your old customized system from a year back. And so you can imagine everything there is what lives in the NPPC deployment.
Cool.
Yeah, do you guys use the event egress to actually in your ribosome? Do you guys use event egress to sync the state from the Flyte execution to your local control plane? I'm going to call the Latch your own Kubernetes deployments as your control plane.
Yeah, we actually don't. We have a tunneling instance that listens to the Postgres deployment,
Right.
and then we have triggers set up.
Yeah. You probably should look at the event egress. That might be interesting, but that's okay.
We'll definitely do. Yeah, it might be better. That was a pain in the ass to set up what I just described.
Yeah, the tunneling is, it's pretty hard. Yeah, I had a question about this actually. Sorry, we are going a lot into the security aspects, but it seems that you probably encode the nucleotide, as you said, in a string, and so if from our point of view, if Flyte admin is the one that actually stores the primitive value, which includes a string, does that and this is for Greg and Jay and whoever's here who are deploying their own Flyte instances. If the UI shows that value, is that desirable or is that not desirable? Because I realize that there's a security vulnerability, but there's also UX and it's a constant fight between them. And what is the right experience? Does that question make sense? Or maybe I can repeat it again. So like strings, integers, and so on, that Flyte actually shows in the UI today as an input and output of tasks and workflows and so on. Is that a security vulnerability at some level? And if not, why not? And if yes, what should we do? Should we like just an RBAC role enough?
Yeah, I mean, just quickly from our perspective, we don't see it as a security vulnerability. Our customers don't either. I think anything that's small enough to fit in like a reasonable amount of JSON that you can send to a browser is not something that people are like worried about us holding on to. I think people would be worried about us like having access to their entire genomic corpus, but any sort of like input or output, little string of nucleotides that you display to someone to make them understand, how the workflow did is not something people have expressed concern over. I do imagine though that like technically and maybe longer term, we would have to figure something out, both you and us.
Yeah, we do have some ideas around it. We'll definitely love to share with you guys also what we're thinking. So, cool. Any other questions? If not, like I think the Latch team is just fantastic. They're rock stars. They just go in, solve the problem, get things done, and I'm just amazed at how much they get done in like just a couple months. So, all power to you guys, and thank you for supporting us. And thank you for sharing.
Really appreciate that. Yeah, and like I said, we really do believe in Flyte. Like there's no marketing incentive here. Right? It's something we just believe in as a technology. So we'll continue to support and contribute and work with you all. We've learned a lot from your team through the PR process, and we're excited to keep on doing that.
Oh, thank you, Kenny. Thank you. Yeah, it's been a long review, but like I think I'm so excited about all these features. So thank you for contributing. And for everybody else, like it's okay to contribute. It like I've actually had some people ask me questions that, hey, I've never written code in this kind of an open source project. What should I do? It's okay. Open up a PR. But then be open to like getting comments and we'll work with you. I think the core maintainer community is very, very open to receiving new code. They understand that it's not always easy to contribute, but we also are obliged to maintain quality of the code for a large community. So, but that doesn't mean you should not. Please open up, and we'll work with you.